So first thing is when you're saving your data, always save it under the raw file extension. So if you're working on a Leica system, that would be .lif or .lei. If you're with Zeiss, .lsam or .czi, etc, etc. Now the reason we do this is because it doesn't just save the image, but it also saves what we call metadata. So information about exactly what the microscope settings were on that day when you took that experiment. The other nice thing about this is a lot of the softwares also allow you to reuse those settings again on another day. So you can be imaging the exact same way many days, weeks or months apart. Second thing, if you're going to export your data out, then please export it in the TIFF file format, because this again keeps the original information for any downstream quantitative analysis that you're doing. The issue with using things like JPEG, GIF or PNG is that very often they use what's called a lossy compression, which actually means that you lose information. So if you can see the two images here, the one on the right is kind of a bit more averaged out, so there's a little bit less information going on in this image than there is in the TIFF image. So you can still use these for things like PowerPoint or for printing things out for your lab book. But if you're doing any quantitative analysis, then we recommend you save the images as a TIFF file. Okay, number 3, modifications. So never process or modify on your raw files. Always make sure that you keep a copy. And for every modification you make, save a different file. Have something in the file name which makes it obvious what modifications you've made at that point. Someone should be able to come in, look at your data and work out what steps you've taken. Next thing is make sure that you're backing up your data. So your supervisor has signed an agreement with the funding body who funds their research that they're going to ethically look after their data. So they've got an agreement saying that you're going to doubly back up your data, you're going to keep data for something like 5 years after publication. So don't ever use the microscope computer as a safe place to store your data. There are instances where there are hundreds of people accessing those computers and they can either accidentally or deliberately delete that data off. I also suggest that you don't keep all of your data on an external hard drive. We've had a case in the past where someone's lost their hard drive with 6 months worth of work on it, and the only way of getting that work back was to go back and do all that work again. You don't want to be in that situation. Okay, next thing is, as I've already mentioned, although it looks like an image, it's actually an Excel spreadsheet of values, so your image is actually scientific data. So if you're trying to make the image look more beautiful, you're actually manipulating the data. Now, hopefully nobody would go into an Excel spreadsheet and start randomly changing numbers. And you can no longer get away with saying, oops, I thought that'd be okay, or I wasn't aware of this. So you really have to be careful and treat your scientific data ethically. And we'll take you through a few examples over the next few slides of what you should and shouldn't be doing. So in this example, on the left-hand side, we've got an original electron microscopy image, and on the right-hand side, we've got a manipulated image. So in electron microscopy, you can detect proteins of interest using an antibody, which has an immunogold attached to the antibody. And in this case, I guess these researchers think that these larger blobs here are their immunogolds. They've also got 2 species with different sizes, 2 different nanogold sizes. So they've gone in and they've increased the contrast on these structures that they think are their nanogolds. And they've got this weird blob going on here, so they've gone in Photoshop and taken that out. Hopefully you guys can see that that's not ethical. They're making specific modifications to specific parts of the image. Next example is a fluorescence microscope image. In this case, we've got a group of cells. The image looks reasonably okay to me. There's a little bit of saturation in some of the points. But if you do a simple contrast adjustment, you can see the cells in the bottom left-hand corner and the group of cells in the bottom right-hand corner have a different background to the rest of the image, which suggests that they've been cut and pasted in from another image. So again, this is obviously not ethical, but it's a very easy step to do this type of analysis, and a lot of the journals now have editors whose jobs are going through and looking at this in any submitted articles. Again, not ethical. Next one is modification on a specific feature. So in this case, we've got a fold up here in the left-hand corner on the image, and that's been taken out in the manipulated image. So again, not ethical. We're making a change to a specific feature within the image. There are a few ways we could get around this. So if the fold isn't sitting in an area which is important for the interpretation, we could just leave it there. It's always nice to see some realistic data in papers. Again, if it's not important to the interpretation, you could just crop it out and have a square around the region that is important for the interpretation. If it's vitally important, then you can always go back to the block and section out and try and get a section without a fold in the important region. In this case, in our original image, we've got a bit of a shaded area in the bottom right hand corner here. It doesn't look like this microscope was aligned properly. And in manipulated image, you can see that's been cleaned up. So first thing is make sure your systems are working properly. If this is a bump microscope, for example, please let us know if you're seeing issues like that, and we'll try and sort them out for you. This would be acceptable only if no changes occurred to the actual tissue, and also a statement is made in the figure legend. Now, I would say that that's a given for any manipulations that you make. You must tell the reviewers and anyone reading exactly what you've done to the data. Last one is we've got our original image on the left. There's a bit of uneven illumination again, and we've removed all of this background tissue as well. Again, not acceptable. We're taking a large degree of background alteration, even if it's clarified within the text. Unethical. Okay, I quite like this experiment. So we've got a sample, which we're obviously interested in. We've got a positive and negative control, which hopefully have been used to set up the acquisition settings. But again, if we do a simple contrast adjustment, we can see that our negative control is completely black. Now, you will never get a completely black image off a microscope. You'll always have some kind of electronic noise from the detector, which contributes some intensity. And you can also see the background between the positive control and the sample are also different as well. So it may be that these images have either been acquired differently or have been treated differently in terms of visualization. So again, not ethical. So what can we do? So we can make a modification to the whole image. So we go back to the intensity graph in this case, and in this case we've got an example of some data taken where we haven't used the whole dynamic range, or majority of the information is down in this bottom left hand corner here at the low intensity range. So what we can do is increase our maximum intensity down towards where the data actually is to make this a bit brighter for visualization. So again, I should stress this is visualization only, not quantification. If you do this and hit apply, you're going to rescale all of this information across the full range. So here we've rescaled it. This is our lookup table showing a saturation. We don't have any red pixels here, so we're not hitting the data, we're not saturating any of the details. Another example, if we go too far, so we've gone beyond where there's actually data, now we can see saturation. And if you compare the structures down here to the ones above, you can see they're actually a lot larger, and that actually changes our interpretation of the data. Okay, so you can adjust up until the point where you're starting to hit information but not beyond because we changed the interpretation of the data. So that's misrepresented. Okay, so just to clarify, all image modifications must be ethical. We can adjust the minimum and maximum positions for visualization, not for quantification. Do not just adjust gamma. So a lot of the times on these intensity graphs you see these lines. This middle position, if we were to move this up or down, is switching what we call gamma, which is a non-linear adjustment. Again, it's probably best if you avoid that if you can. And if you do make any differences, then please be descriptive in the methods sections of your publications.